I'm going to introduce the first speaker of the second session after the break, and Dr. Babak Alipanahi. Babak is a computational biologist and machine learning scientist with over a decade of industry experience. He is the co-founder and chief scientific officer at Exai Bio, which is a company that I admire highly. Where they're delivering next-generation liquid biopsy platform for the earliest detection and most accurate cancer diagnosis. And prior to Exai, Babak served in research positions at Google and 23andMe and co-founded Deep Genomics. Babak has been applying machine learning algorithms to solving biological problems with an emphasis on disease detection and etiology. As a postdoctoral fellow at the University of Toronto, he won the university's prestigious Invention of the Year in 2016, an award for his research on using deep learning for modeling biological processes. He has a PhD in computer science from the University of Waterloo in Canada. Thank you so much, Babak. I'm very excited about your talk.
Thanks, Homa, and thanks to the organizer for this opportunity. So Homa made my life easier. Thank you so much. So let me just focus on the third line. I did a research internship with Microsoft Research in Cambridge in 2010. And that has been, one of the highlights of my life. And I hold Cambridge very close and dear to my heart. And I hope that, one day I can go back and stay for longer. Many, many good memories. Microsoft Research was in J.J. Thompson Avenue, and it didn't have a cafe. So we would dine in the Cavendish building, in the Cavendish cafeteria. And yes, a lot of very fun memories. And it was like a pivotal moment in my life because Chris Bishop, who is one of the pillars of machine learning, changed the way I think about AI and think about application of AI. Okay, so with that, what is a hyper-SVM? It's funny. So when I joined the University of Toronto in 2011 as a postdoc, we were thinking on modeling splicing. So here, psi-ti is inclusion of exons in tissue T in gene I. But what matters is that when we designed this very simple idea as a neural network, as a probabilistic neural network that you would inference, do inference with Bayesian modeling, our collaborators were not keen on using the term neural net. So hyper-SVM is some joke that Brandon Fry, my advisor, came up with. So I'm coming from a time that's actually having the term neural network or machine learning in your paper was a good reason for it to be rejected. We have come a long way, and now Ilya Sutskever is on the news, and this is right along the time that Ilya, Alex, and Jeff Hinton were working on the ImageNet, and everyone was like, "Oh, they're buying these GPUs, yeah, for sure, for deep learning." Anyway, we have come a long way from them, and from hyper SVMs to chat GPTs and DALL-Es of the world. Okay, so cancer is one of the most challenging conundrums of our modern life. We are living longer, we have controlled infectious disease, we have vaccination, yet cancer is a huge problem. And honestly, I don't think cancer can be defeated anytime soon. And by cancer, I mean advanced cancer. So our best treatment for cancer is early detection. So here you can see the 5-year survival rates for cancer patients whose tumor has been diagnosed at different stages. Between stage 1A to 4A, there is like 9-10x difference in survival odds. So we really have to detect cancer early if we want to significantly improve outcomes. And what we do at Exai Bio is that we combine the richness and the information in cell-free RNAs with AI to reinvent liquid biopsy. Okay, so this is what happens for those who don't know. So on right, we have a tumor, and these tumors, they die and they shed some of the contents in extracellular vesicles, EVs. And in these EVs, we have RNA, DNA, proteins. But at XI, we focus on RNAs because they are stable and they are released from tumor cells. Cell-free DNA, in contrast, it needs tumor cells to die, and then it's released into the extracellular environment. Tumor-derived cell-free RNA is detectable in blood. It's very information-rich. It can provide a window into gene expression programs inside the tumor. Also, a large number of orphan non-coding RNAs can be detected in cell-free RNA profiles. So what are oncRNAs? So the classical view of cancer is that you have normal cells, and through changes in expression programs, they morph into cancer cells. This happens by underexpression of tumor suppressor genes or overexpression of oncogenes. And we are all familiar with this. But at XI, with the science that came out of Hani Gudari's lab at UCSF, we are interested in new emerging RNA species, RNAs that are generated because of de novo transcription of newly open chromatin, or because of aberrant post-processing of RNAs that results in new RNA species. So we have tens or even hundreds of millions of random RNAs in tumors, and we are not targeting all of them. Our focus is on those new RNA species that are only found, in a significant manner, in a replicable manner, and can be found in multiple tumor samples. We call them orphan because, borrowing terminology from bacterial genomics, meaning that they are not observed in the mother cells. So the thing is that these form signatures. OncRNAs are signatures. So in this heat map, every column is one oncRNA and every row is a tumor sample from a different patient. Here you can see that, if you squint your eyes, you can see as if each cancer has a barcode. So not only you can use oncRNAs to detect presence of cancer, but also you can identify the primary site, identify the subtype. So at XI, we have looked at many different cancer types and different modalities, but let me talk about NSCLC. It's the #1 cancer causing deaths in North America. And surprisingly, I think, Amin also alluded to it, the compliance for a low-dose CT scan in California is 1%. This is not for general population. People who have smoked a lot of cigars and smoke for many years, and they are eligible for lung cancer screening, only 1% of those actually undergo screening. And we want to change that by making cancer screening more approachable, more feasible, and easier. So this is how it works. We always start from the tumor. We have hundreds and thousands of tumor samples. We get small RNA profiles from them, and we compare it with the RNA profiles of healthy lung tissue. And we have our own pipeline. We identify a set of candidate oncRNAs. We have an independent non-cancer reference set. We use it to make sure, we are not adding any RNA that has bona fide function in a healthy body to the oncRNA set. So this gives us the fingerprint for oncRNA. Just for demonstration, assume that the pattern for lung NSCLC is digit 8. A black pixel means that oncRNA is not expressed and observed in lung cancer, and a white pixel shows that it actually is expressed and profiled. So at the test time, this is what happens. We drew blood from patients. We assayed small RNA profile, and then we form the oncRNA profile of the patient from blood. Here you can see that many pixels are green. This is because we can't observe them. This has many reasons. One of them is that, for ethical and practical reasons, we cannot get a lot of blood from patients. Remember, tumor sheds into blood. We have 5 liters of blood, but we drew, just a few cc's. Also, we have limited sequencing depths. We cannot sequence to exhaustion. So a mil of serum has 1 to 10 billion RNA molecules, and in a good day, we can assay 10 to 25 million of them. And also, like, some oncRNAs are not released to blood. That's another thing. So that's why we need an AI model to compare the fingerprints, the barcode we have generated from tumor data with what we get from patients and reconstruct the oncRNA profile inside the tumor and make predictions based on it. And why we need an AI model? Because this is a really hard problem. And I mean, many people are surprised that the hardness, more than coming from the fact that, the tumor or cancer is complicated, is because getting biological data is hard. For example, you have to get data from different hospitals. And each hospital, each clinic, each nurse has their own technique, uses their own needle type, uses a slightly different tube type. And these effects combine and make training models extremely complicated. So we have a large cohort, it's 900 almost 900 subjects, 320 NSCLC cases, 567 individuals without cancer. I have to say that, when we say without cancer, 1% of these people probably have some neoplasm, some sort of lesion in their body. That's why this is not the ideal cohort, but it's good enough for our purposes. We are enriching for early stage cancers because that's when, as we talked, it really matters. And this is data from tens of hospitals, sites, countries, from Germany, from Ukraine, from Russia, from Venezuela. We are trying to diversify the input so that, the model can generalize. And, one thing I want to talk about is that as someone who has spent years and decades on developing AI models, I will be the first person that would push against, overhyping and inflating our results. One problem that I see rampant in our field is that baseline models are underappreciated. So when my team comes to me and tells me we have to use this model that has this complexity, I always tell them that you have to show me that we have done every effort to use a simpler model and it has failed. And here, on left, you can see the ROC curves for a linear model. And, we have trained the best possible linear model with best regularization. And we're comparing with our, Orion AI model that you can check our website. We have in our publications, and the paper is coming out soon. And we see a real drop in performance. So the figure in middle, let's spend some time on it. We have sensitivity of diagnosing cancer for different stages. So, I mean, it goes up by stage as we expect because later stage tumors are larger and shed more nucleic acids. But it's great, at stage 1, we can identify almost 90% of cancer cases at a specificity of 95%. So this is a test that you can take when you go, do your cholesterol check or your, kidney function checked at your annual physical, and it will have with some cancer screening as well. I mean, these models can never be super accurate. That's why there's always going to be a follow-up, but we are reducing the people who need to have a follow-up by a factor of 10 to 50x. And lastly, we are at XR moving beyond detection. So detection is just the beginning. What's next? What's the subtype? Is it an aggressive tumor or not? Should it be treated immediately? Does it need surgery? So these are all the questions that we are trying to answer at XI. One of them is to subtype the tumor. And here we are really proud because, using a blood-based assay, we have been able to subtype the tumor, the lung tumor, almost as good as what pathologists would do looking at the slide from the actual tumor. And, let me finish with these two notes that, nothing is as humbling as biology. I have worked with, a lot of great people, and this is something that this motif I've heard from a lot of my mentors that, a good result is just a beginning. You have to justify it. You have to make sure, there are no major gaffes, major bugs. There was a paper, that they had amazing performance in detecting, I think, lung cancer from radiology graphs. And it turned out that, the clinic that screened the cancer patients had their logo printed on the bottom left of the graphs, and the AI model was just, detecting the logo. So we have to be really careful. These AI models give us a lot of flexibility, a lot of power, and with power comes the responsibility. And as you know, it takes a village. I think around a third of our people are not here because we pride ourselves in having a hybrid work environment. We have exonauts in Canada, in all states in US, and really want to create an environment that people can do AI for good and biology for good. And with that, thank you so much. And I would be happy to answer questions offline if there's any need.
Thank you so much, Babak. That was excellent. And thank you for keeping the time. I think Nishant will be asking questions here for the second and third session.
Yeah, we don't have any specific questions from the audience. So just a reminder, if for all our attendees, please feel free to ask any questions away. I'm not in the world of biomedicine, but I think a lot of people are definitely talking about the GLP-1 drugs. So I was just to see, because obviously there's speculation that perhaps those class of drugs could be used to cure diseases like cancer, but just curious to hear your views as well on that topic.
Yeah, I mean, those drugs are actually not new. They are like newly, not even discovered, like newly went into fashion. No, I think those drugs, like for some people, those can be life-changing. And you are right, one of the biggest factors for cancer is obesity. People who have type 2 diabetes, I don't remember the exact number, but I think it's 5 to 10x higher chance of pancreatic cancer. So obesity causes inflammation. Inflammation promotes cancer. So I agree. I think those drugs, Wegovy, Ozempic, they can be life-changing for a lot of people. But before we go to a place that we need that, maybe we should, be more cautious about our diets, be opportunistic vegetarians and vegans, exercise more. But, we are mostly, millennials and Gen Zs here. I would say the cancer among the youth is skyrocketing. The rate of colon cancer, late stage colon cancer today in people under 40 is 5 times higher than in 1950s. So Dana-Farber has a new clinic for colon cancer for young patients. And this is not a screening artifact because, people under 40 are not eligible for screening. So it's not that we are screening more. We are eating more unhealthy. We have more worse life habits. We are eating, ultra-processed food. And I think we all need to step back and, think about the long-term impacts of, what we eat and how we go on with our lives.
Thank you, Babak. I did not know that. I personally have a lot of questions that I will ask you offline. Thanks again for giving us some of your time and sharing your data with us.
Sure, absolutely. Thank you.